Thank you very much, Philip. I guess I take it as a compliment rather than making me feel old. So I know it's Thursday. It's day 4 of the conference for most of us. It's probably beer o'clock somewhere already. So we'll take things a bit slower. I'm mostly going to tell you a story. There will be results in my presentation, but they're really not relevant to the story. So really just feel free to relax, and mostly I'll summarize the key take-home messages at the end if you miss things in between. So Richard already made a really good intro of the APPN yesterday, so I don't have to talk about that much. So just want to mention that within the OzBarley project, most of the work was done initially at the Adelaide node. We now also have work at Western Australia and in Wagga Wagga as part of the project. And we're covering everything from the controlled environment to field phenotyping with this project. So barley is a key cereal crop in Australia. It's the second largest crop after wheat, and about 30 to 40% of the world barley trade is Australian malting barley. So tonight, if you're drinking a American beer, that's likely going to be American or Canadian barley. If you're drinking a Mexican beer, there might be a chance that that contains some Australian barley. But really our biggest importer or trading partner for barley is China, and also the Middle East, mostly for obviously feed barley. So barley production in Australia faces a number of challenges. We did have recently some trade disputes where heavy import taxes were imposed, which heavily impacted the industry. They have now been lifted, but we do obviously also have ongoing environmental fluctuations, and they are not getting any better with climate change. And then growers obviously face the constant issue of heavily fluctuating prices, which can massively impact how they can market their grain that they produce. And also further downstream for the brewing industry and maltsters, they are a very electricity and water heavy industry. So there's obviously also the challenge of reducing the environmental footprint there. So we've heard a little bit during the conference about various germplasm collections for sorghum, for example. Harkemal mentioned one of the rice diversity panels. And we got together as a group and discussed the lack of a barley germplasm collection that sort of represents what's going on in Australia over the years, and also something that could be used by the industry. So we got together a large team, and I'm putting this slide here at the beginning so I can thank everyone involved rather than potentially forgetting something at the end. So we did have an opportunity to get funding to get this project off the ground. Interestingly, that came partially from the ARDC, which is the Australian Research Data Commons. So you wouldn't necessarily think that a facility that's mostly focused on creating data infrastructure would invest in a barley project, but it was really about generating a data asset that meets the FAIR standards that could be used by the community. And then APPN obviously co-invested. Bioplatforms Australia is our sister facility, so they heavily focus on the genomics, proteomics, and metabolomics side. And more recently, the Grains Research and Development Corporation invested in MEL. We also had a number of public-funded research institutions mostly contribute their time and goodwill. Importantly, wrong button. Most importantly, the Australian Grains Genebank was involved right from the start. And also we had all of the 3 major commercial barley breeders in Australia on board, and they contributed heavily in terms of their in-kind support. So how did we go about creating the OzBarley panel? We pretty much put out a call to the community. We talked to the breeders, to the main research groups around Australia to come up with a wish list of all the barley lines or cultivars that had been released over the decades of barley breeding in Australia that should be included. We then sourced the material primarily from the gene bank, but also some from breeders and other contributors. Peter mentioned that material in the gene bank can sometimes be heterogeneous, which is perfectly normal. So we went through 2 rounds of single seed descent to create genetically pure material, and then bulked up the material in the greenhouse. In parallel, we also did, mostly Julian, did an awful lot of detective work trying to piece together this very complicated, pretty much pedigree of Australian barley breeding. I don't expect you to be able to read anything here, but just to show you how complex pedigrees can be in breeding programs. And the ones you can see at the bottom, they are lines that are currently grown in Australia. What was also important when we put together the panel is that there were no IP restrictions on any of the lines that we were using, so that the material could be easily shared later on. So just zooming in to give you a bit of an idea, there are some lines like Proctor here or Clipper that are highly important because they are the parent of many lines within the pedigree. And then there are other lines that, that are pretty much once off in a sense that they were bred but didn't really feed into any of the further breeding programs. Okay, so we've got the material, we've got the pedigree. We obviously wanted to genotype the material. So in addition to using an Affymetrix SNP chip, we also used RNAseq as an alternative method for sequencing. We didn't have the budget to many different tissues and organs, so we really focused on germinating seedlings because that has obviously relevance for the brewing industry as well. And that was all done by our colleagues at CSIRO and the AGRF, which is the sequencing facility in Australia. So for our SNP chip, we've got about 12,500 SNPs, which is sufficient to give you a pretty good coverage across the barley genome, but it's not really high density. So with the RNAseq, we could then enrich that to over 100,000 SNPs. And you can see here, for individual genes, we have quite a number of SNPs. Obviously, RNAseq is limited to transcripts that are present. So this gives you an idea of what the results may look like. So at the very bottom, we have the MOREX3 reference genome. We also have MOREX in our collection, and thankfully there is not a single SNP in that region, as you can see. It's always nice when your version of MOREX turns out to be the same as the reference genome, which isn't necessarily a given. Similarly, CM72, which is another 6-row barley, again, no SNP in this specific region, whereas Wimmera, you can hopefully see those vertical lines. Each of those represent a SNP that is present here. But then we also have the case where 2 of the varieties don't have the gene expressed at all. So we do get quite rich information from this type of genotyping. So we have the pedigree, we have the genotypic information. How do they compare? And obviously with genotypic information, the classical approach would be to do a phylogenetic tree. We tried something slightly different. just because of the way the relatedness was between the lines. So we created a net that in a sense shows you the distance of different genotypic difference between the lines. You can see here, that's mostly where some of the European ancestor lines cluster. Down here, we have some of the North American ancestor lines. Over here, some of the 6-row Asian lines. And then in this cluster is a lot of the modern Australian cultivars. In addition to the landraces, where we've got about 220 lines, we then also to the elites, we then also added landraces to the story. And we thought if we do a PCA on the genotypic data that we would get nice separate clustering of the two. That wasn't the case. So you can see that the landraces and the elites cluster together for large areas. But we do have some clusters that are, in a sense, landrace only. We were trying to select landraces that have a broad geographic spread and are sort of semi-dwarf. They weren't all that dwarf when we grew them in the greenhouse, so they obviously come with big challenges. As Peter already mentioned, dealing with this type of material is not like dealing with all the elite ones. So we've got the genotypic data, we've got the pedigree, we've got the pure seeds. So we then proceeded with phenotyping, and let me see if this works. Yep. So we did sort of a baseline phenotypic screen in our controlled environment facility where we literally just wanted to look at growth over time. We also harvested spikes at the end, put them through our X-ray scanner so we can look at spike architecture and seed morphology. And you can see some lines that are a lot earlier flowering, like unicorn, for example, that's represented in the growth curve peaking earlier, versus some of the sort of later developing lines like dictator further down. Okay, so we've got genotypic data, phenotypic data. We can obviously do GWAS. This is really just a simple one for looking at row type. And I'm not going to go into any of the loci or anything. That's really just an illustration that we can do that. With the data we've got, this is work from one of my PhD students. He's interested in root growth angle. Now, in most places around the world, you are looking for deep roots with a fairly shallow angle. South Australia, we don't have great soils, so mostly what's down there is toxic and not much water, so we're actually looking for a shallow root system. So more like the one on this side here. And again, we can use the phenotypic data, combine it with the genotypic data to then do GWAS. Okay, so how do we put it all together? We've got genotypic data, pedigree data. We've got some basic growth analysis, some basic phenotypic traits like height and other aspects. For the elite panel, more basic phenotypic for the landraces. And then we were looking for a database where we could upload all this data and make it available. And that's where we then realized that such a magic database does not exist. So we've used a bit of a hybrid approach. And when I say a bit of hybrid, you can see that it's quite fairly hybrid. So we have a landing page on Figshare for the sole purpose that allows us to mint a DOI, which we obviously want to use for tracking usage of the resource. We are using GERMINATE, which is a software developed by the James Hutton Institute that can host the genotypic data, the pedigree data, some very basic phenotypic data as well. The large numbers of image data is too big for GERMINATE to handle at this point in time. So the raw imaging data is hosted at PAWSI, which is a data storage facility funded by the national government in Australia, similar to APPN. So that's where the raw image data is hosted, and there are hyperlinks from GERMINATE to the respective data entry at PAWSI. The RNAseq data as well, the raw data, really not suitable for GERMINATE at this point. The SNP data extracted from it will go on to GERMINATE, but the raw data is on SRA. So that gives you the infrastructure. Now, what does GERMINATE look like? We haven't published the paper yet, but if you know how to use Google, you'll find it. So this is pretty much the landing page. Gives you information about the germplasm, the markers, the traits, and location of the material that is in there. You have a menu over here where you can apply different filters and search terms and so on. So here's an example, and I don't expect you to read any of this, but I just filtered for a trait, which is peduncle length. So the peduncle is the bit of the stem that is pretty much this bit here between the last node and the spike. And you then get a box plot for the entire data set. And you can even click on individual dots, and it then pulls out which variety that is, or you can download the data in CSV format from GERMINATE. So if we then click on one of those examples, so we then get the passport information for GERMINATE. We do have a pretty good passport completeness index. We had a very dedicated research officer spending a lot of time trying to get this passport score as high as possible. And we'll soon add DOIs when we deposit them back into the gene bank, so that'll lift it even a bit further. You can also have a look at the X-ray images, so projection of the individual seeds, or a slice through the spike for each of those entries. And we then also have a link to the raw images. So these are hyperlinks where you can then click on and get the raw images that were collected during the growth analysis. So this is all controlled environment phenotyping. Obviously, barley isn't grown in a greenhouse for production. So we also wanted to look at traits and phenotyping out in the field. And we took a bit of a different approach for this, which is what I'm going to call community-based field phenotyping. So we had funding from the GRDC to run trials. So we've got 3 trials out at the moment in Western Australia, South Australia, and New South Wales. And we'll have another set of 3 trials out next year. And what we did is we put out an open call to researchers across Australia, everyone from PhD student, early career researchers to senior academics, to say, we are going to run these trials. If there's a specific trait that you want to phenotype, you can come and access the trial, as long as it obviously doesn't destroy the trial. We also had some travel support for students and EMCRs. So what we did, put out this call, we got applications in, and there were quite a number of different traits that people were interested in. Some wanted to look at spike architecture. Some wanted to look at head loss, which is a big agronomic problem in Australia, where entire heads snap off, fall to the ground, and are obviously lost to the farmer. Lodging can be a problem. We have salinity widespread throughout Australia, so sodium and potassium accumulation can be an issue. We also have a PhD student who wants to look at things like nutrient use efficiency. So the photos you can see here are pretty much from this year, where researchers went out to do measurements. Some accessed just one of the trials. Some were accessing all 3 trials across the country. We will also have yield data from 2 of the trials. South Australia, unfortunately, didn't get any rain during the critical period, so that one will probably turn into chicken feed, which is rather sad. But yeah, what you can see here is us sampling leaves for salinity tolerance measurements. So we're looking at sodium accumulation, very cheap trait to measure. And these are traits where researchers look at photosynthetic traits at the trial in New South Wales. And what that really allowed us also was to connect researchers with each other. So we had applications coming in from someone in Western Australia and in New South Wales, both wanting to look at photosynthetic traits. So we were then able to connect them up and say, hey, you're wanting to measure the same thing. A, you can probably help each other out so you don't double up on work. But B, likely to have good opportunity for future collaborations. So with this, I just want to summarize that I believe that working across the omics areas can create a resource for common use. And it can also reduce the barrier of entry especially for early career researchers or researchers who don't come from labs with massive funding. So as an example, this is Harley. He's an honours student. So honours students in Australia, they have about 9-10 months for their project. So there was no way he could create or collect a germplasm resource, get the genotyping done and phenotyping, because of the time, but also because of the cost involved. But by having those field trials out there and all the data publicly available, he can pretty much go out to the field and simply collect the leaves, do the measurements back in the lab, and do the GWAS analysis later on that data. So I think investing in a national data asset like this can also create new collaborations. As I mentioned, we were able to connect researchers who were working on similar subject areas that didn't necessarily know of each other's work. And with that, I just want to say thank you to our funders and to you for your attention. And I'm going to leave you with a photo of the OzBarley trial in Western Australia, which was taken about 2 days ago. Thank you.
Thank you very much, Bettina. And the room is open to questions. Rick?
I think there's a prize for asking the most questions. I'm on it. Now I was thinking about, I'm going to ask a silly question here, but the roots, right? You were selecting the phenotypes that have shallow roots because of the soil that is toxic. Yep. Apparently your soil is only 30-50 centimeters, I once learned. Aren't the roots smart enough that if they hit the toxic zone that they simply decide to go shallow?
They might, they will likely divert eventually, but there is one aspect which is growing into toxic soil. The other aspect is, in a sense, wasting resources by potentially growing into soil where there is no water. South Australia has a so-called Mediterranean climate, so we have intermittent rainfall. So the shallow root system actually helps to capture that intermittent rain that comes during the season. So having that shallow root system right from the beginning will help plants to then capture that precious bit of rain that comes most years, not this year in South Australia, but thankfully it did come in the other states.
Okay. Thanks.
Yeah, very nice talk. I'm interested in this community approach. I think that's a very good idea. But are there any, did you select people, or could everyone join for every type of research, or how did you organize that?
So I guess the restrictions we had was the trial had to remain intact, so we couldn't allow someone to do, biomass cuts halfway through the project, because that obviously would then ruin the trial for others who want to do harvest traits, for example. The other aspect is that certainly for the ones who receive travel funding, there is a condition that after a quarantine period where they can work on their data to publish, that data then becomes publicly available under CC BY4 license, like everything else in the OzBarley project, and then becomes part of the GERMINATE portal as well. So really the idea is that we funded the creation of the resource. We had funding to run the trials, and people can access it, but the expectation is that they share the data to then grow the overall data asset for others to benefit as well.
And were there any things that were not studied that you hoped that people would come for analysis, or did you have the full spectrum of things that you wanted to acquire?
I think there are always more things that you would like to measure. So we probably didn't have much metabolomics or things at this point. We will likely have someone looking at proteomics of the mature grain at harvest to look at protein content. Some people wanted to look at disease tolerance. That was one thing we couldn't facilitate because these trials were managed according to best agronomic practice, so we couldn't run a disease nursery. But yeah, that is where the limitations are.
In the back.
Thank you, Bettina, for sharing this nice resource. I just had a question on the data set. So the RNAseq data for this panel, is it sparse, that it's only good for SNPs, or you could actually do functional analysis at expression level?
We didn't have the resources or the financial resources to do multiple tissues, and we also couldn't do replication. So it's really mostly for, I guess, presence, absence of expression. And SNPs, it's not good enough to look at expression levels.
Thanks.
Yeah. Great talk. Do you have consideration to integrate the barley ontology into the data platform? There is a trait dictionary existing for barley.
So the traits that we measured, sort of the existing traits, things like, height or yield or sort of established phenotypic traits, we try to use ontologies as much as we possibly could throughout the project. There were some traits, obviously, that we measured with a phenotyping platform where we had to create our own vocabulary. Again, for this, we tried to use best practice. So we worked with people that work in the ontology and data vocabulary domain to have that as sort of consistent and standardized as possible.
So you have the ontology in the GERMINATE application. Is that what it is?
Yeah, so the traits that we measured, where there is an existing ontology term, we would use that ontology term, yeah.
Yes.